In the fifth chapter of medical school pathology, we will be dealing with the general topic of genetic disorders. Of course, understanding that every disease, like every cell, has genetic counterparts. Nevertheless, there are still some diseases which are regarded as genetic by virtue of a variety of factors. Uh, I want to mention that genetics and especially the recent advances in the last several years in DNA and gene splicing and so forth have just completely revolutionized medicine. And uh, I can really say that almost everybody that graduates from medical school within a few years, sometimes less than that, feels terribly inadequate when it comes to gene topics. It's probably the only real branch of pathology or medicine in general that it has a massively exponential rise in understanding and discoveries compared to all the others. Nevertheless, there are some classical things that are regarded as fairly constant. Genetic diseases, like I say, is just about every disease because every alteration in the cell is ultimately the result of its genes, its DNA. Some diseases we can understand as being classically environmental and relatively independent of genes, like for example, gunshot wound. Uh, in reality, most diseases are a combination of both, and let's keep it at that. Let's talk about mutations because we know that DNA mutates. The classical type of mutations which result in patterns which we will call diseases can be of three types. They could be genome mutations, which involve problems regarding a whole chromosome, for example, lack of one or an extra one. Chromosome mutations are usually visible through the routine types of chromosome banding that we do, and they're more in the realm of things like, deletions, translocations, things that happen on individual chromosomes. Many mutations, or classically mutations, are the result of gene mutations. And in many cases, this could result from a single base pair error. In many diseases like that, classically the lysosomal storage diseases, and of course the prototype hemoglobin S or sickle cell disease is the classical example. A gene mutation can be a deletion of a single nucleotide or a substitution of a single nucleotide. If you kind of visualize your double helix, well, just knock one of those little nucleotides out or substitute it with something that shouldn't be there. So here we go. We have our DNA. Ultimately, it's going to be transcribed to be messenger RNA, and then it will have your translation down at the ribosomal level. The classical understanding of single gene diseases would be in sickle cell disease. There are beta chains in hemoglobin, and in the beta A, which is normal beta chain for hemoglobin, you have A. Does that sound like adenine? Yes, it is. Well, in the disease which causes sickle cell disease, this adenine is substituted by uracil. So rather than having a GAG, which codes for glutamic acid, you have a GUG, which codes for valine. And on the basis of your beta chain having one single wrong amino acid, in other words, point mutation, on the basis of one single wrong trinucleotide sequence, you have a disease which could be very, very, very serious in the heterozygous state. We talked about point mutations in sickle cell in which valine, there's a valine glutamic acid substitution. You can also have mutations in non-coding portions of your DNA, parts that are very much involved with transcription. So a mutation in a non-coding sequence, which doesn't code for a specific structural or functional protein, might result in a defective transcription process in terms of taking your DNA and converting it to a messenger RNA. There are deletions and insertions called frame shift mutations, and often this involvement is not in a multiple of 3. There's a classical disease called Fragile X syndrome on the X chromosome, in which you have trinucleotide repeats many, many, many times. Something goes haywire, and for some reason the nucleic acid starts repeating CGG, 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 resulting in a protein which is then not made, which is critical in neuron development. So these are the classical ways we understand gene mutations. Gene mutations, if you think about it, can interfere with protein synthesis. If you believe in the one gene, one polypeptide, or one protein theory, we also said they can suppress transcription from DNA to messenger RNA, or they can produce an abnormal messenger RNA on the basis of the fact that there was an abnormal DNA. These defects can carry over into translation, into protein manufacture level at the level of the ribosome, or they can produce abnormal proteins without impairing synthesis. So all of these possibilities are common, and not only are they, well, all these possibilities are possible, and not only are they possible, but they do happen. Genetic disorders can be thought of as being single gene mutations following the classical Mendelian inheritance patterns, which is going to be a very good fraction of what we'll talk about in this chapter. Or they can be thought of as having multifactorial inheritance, multigenic, multifactorial. And the best way I could give you an example of that is for you to understand that your protoplasm is a result of your genes, many genes. And if you develop a disease on the basis of that protoplasm, you have to remember, if that protoplasm is soil, your relatives are going to have similar soil. So I don't think soil theory is anybody but my own, but that's the best way I can understand it. Then we're going to be talking about diseases which are the classical chromosomal disorders, either defects of chromosomes or additional chromosomes or absent chromosomes, identified by a wide variety of techniques. Okay, let's get into the classical Mendelian inheritance patterns. I think we already understand what an autosomal dominant disease is. It's a disease that can be present if either allele is present. In an autosomal recessive disease, the disease is present only if both alleles, homozygous, are present. In autosomal dominant, heterozygous conditions produce disease. And of course, in the classical sex-linked, which is generally regarded as recessive, these are changes involving only the X chromosome. And we're going to give examples of the diseases, maybe talk about them a little bit. We're going to talk about the general patterns that are specific to these 3 different types, and we're going to show trees, pedigree trees, which are classical for all the types as well. And we'll be doing that in the next 10-minute clip, and I thank you very much.